most difficult phase of our preparation, revision, the last lap of our preparation, revision has come. In the next 15 days, probably CSIR net is going to be conducted, and we all have to gear up. We all have to be ready for this amazing odyssey, the competitive exam which decides our fate, but at the same time opens newer avenues for all of us. Good evening, namaskar, sat sri akal, adab. How are you all? Welcome to Biotecnika's yet another powerful, engaging, and expertise driven webinar on CSIR net advanced last minute revision tips and tricks. My name is Shekhar Suman, and today I'm going to be a host, and we will kickstart a new session, an enticing, amazing, and expert driven session where we are going to talk about last minute revision. And that means you all are going to have a lot of doubts. 15 days, 13 units plus part A, heavy challenge. Isn't that right? So our experts have decided not to confuse you at the last minute, but we're going to give you insights empowered by intelligent ways of studying and technology, shortcuts which you will love, and some beautiful tips and tricks which will not just help you qualify this exam, but it will boost your rank for CSIR net. I take pride in telling you this, that our experts start preparing a month before the actual event. And today that we have a webinar, our experts have been preparing for this day for past 30 days. And now that today we are present in front of you, we are going to give you solid strategies, solid revision tricks and tips last minute, which is going to change your preparation level and at the same time encourage you to go fight. Now, a few words of wisdom for all of you who are writing it for the first time or probably repetitive times. Remember, the last over of the match is very crucial. You hit a 6 or a 4 or whatever. Last over decides whether you win or lose. So in the last over, remember, CSIR net is not a 100-meter race. It's a marathon. And this is the last lap of marathon. You have to win. So you have to strategize. You have to decide which parts of the day, what you're going to study and revise, and extreme focus. Don't look here and there. Remember, there is a lot of free stuff available on internet. There's a lot of gossip happening. But you are Arjuna. Jiski nazar, not on the fish. Your eyes should not be on the fish, but on the eyes of the fish. Not on the fish, on the eyes of the fish. So that's what you're going to do. You have to pinpoint, you have to stay there on the goal. That's the mantra. Unidirectional focused approach. And many students have told me that they love the quotations which I send you in the morning. So keep looking at that. I'll send you a quotation for the next 15 days. Every day, some encouraging words in the morning. Look at that. That will help you start your day in a much more effective and powerful way. Remember, regrets and grief is not going to help you qualify CSIR net. Strategies, hard work, perseverance, compounding effect. That's the bone of contention. So all the best to all of you. I'll see you at the end. Let's get started with this powerful webinar, passing on the controls to our speakers.
Hello everyone, very good evening to you all. Can you people hear me? If you can hear me, please send a reply there. All right, very good. So how is your preparation going on? I hope it's going well. This is the last lap, and as Shekhar sir just said, that this is a marathon. And have you ever seen a marathon? In the last lap of the marathon, the people will run faster, right? Because this is the time when the positions actually change. So whoever runs with whole motivation and whole proper encouragement, right? So they are the one who are going to win the marathon, because this is the last lap. And I tell you, many people lose their focus in this last lap. So we at Biotechnika, we don't want any one of you lose your focus, and that's why we are here today with some amazing last minute advanced revision tricks. Okay, so let's get started. So, without wasting time, what we have done here is we have divided the units in okay. So our syllabus, as the CSIR syllabus goes, it's divided into 13 units. So it will be me and Nidhi ma'am coming after me, and I will be giving you some tips and tricks about till unit 1 to unit 7, and Nidhi ma'am will cover unit 8 to unit 30. Okay, and this is Tithi here. And hello and hi to everyone once again. And let's get started. So, the very first thing that I wanted to tell you is, in the unit, what are the important topics? Okay, now see, we have not listed down the important topics as our wish. No, we have listed down the important topics depending on what kind of questions come from the CSIR paper. Okay, so depending on the question paper, the previous year question papers, how it goes, that's how we have separated and we have listed down the important topics from the units. Okay, so these are the topics basically which you can never skip in your revision. Okay, so make sure you revise these topics. So coming with unit 1, which is biochemistry, so the important topics will be the various kind of interactions. Okay, the hydrogen bonds, the weak interactions, then the structure of amino acids, then the protein structure, Ramachandran plot, again a very important topic. Then thermodynamics, then enzyme kinetics, again a very important topic. Every time you will see numericals coming from enzyme kinetics. Metabolic pathways, right? Like the glycolytic pathway, TCA cycle steps, ATP calculation in those steps. Okay, then pH buffer related numericals, again a very common topic which often comes. Okay, then lipid structure, nucleic acid topology and structure, as well as nucleotide biosynthesis. So these are kind of the very, very important topics from unit 1, that is biochemistry. So when you are revising, make sure that you take up these units. Now, what will be the revision tricks here? Okay, so what you can do is you can use some advanced tricks, like advanced tools for revision, like flashcards. Now, what are these flashcards? Have you people Do you people have any idea about flashcards? Okay, let me have some replies. You people have idea about flashcards? What are these flashcards? Okay, Padmaja. So have you made one? Yes. Okay, Meera, you haven't made. All right. Small concepts. Correct. Parumita. So what do we do in flashcards is you just take a card here. Okay. So in this, you just write the topic. Suppose we are doing Ramachandran plot. So just write Ramachandran plot here. Okay, and behind it, okay, behind the flashcard, you write the important points on Ramachandran plot. Okay, or you make a flashcard for charge calculation of protein. So there you write the topic here on one side of the card, on the other side you write the important steps of how to calculate the charge on protein. Okay, so for the small topics, or for the topics where you have marked it like you like a small in small points, okay, that you can write at the back. Okay, now don't waste your time in making the flashcards now. Okay, now it should be your revision time, right? So now whatever you study, whatever you revise, write it down in the flashcard, and then again a week or 10 days before the exam, you can just have a look and revise the topics again with the help of these flashcards. Similarly, enzyme kinetics, membrane lipids, which is again a very important topic. Many times question come from this part, so you can make flashcards. Okay, that is one tool. Another tool that you can use are flow charts. Okay, so flow charts definitely will be more useful for you, where there will be like the metabolic pathways. Okay, I'll give you a hint here is our Biotecnika Conceptica flow charts. Have you people seen this Conceptica flow charts? Yes. So, like as you all know, Biotechnica has many advanced revision tools, and Conceptica flowcharts are one of them. So, if you don't have it, you may try get it now, or you can take a hint from here and make your own flowcharts, whatever works for you. So you can see here in the flow charts here, we have divided, this is the amino acid biosynthesis pathway. So the entire pathway is written in one after another. And on the side also, it is written like what all amino acids are forming from here and how the process is going, how many steps are involved there. So all these small things are also written. Okay, similarly you see with membrane lipids. Okay, so what all membrane lipids are, what is their function, what is their composition, everything is mentioned in a single flow chart. Okay, so if you are able to make like this, that is going to help you in your revision. Okay, see, revision is not like reading. Reading is when you study something for the first time, you give your full attention and understand the entire concept. Revision is will take lesser time, right? It is revision. So it's just to give enough information to your brain so that it can ignite the memory, and that you will be able to recall it. Okay, and faster is the trick here. Revision should be fast, it should not take as much time as you are reading it for the first time. Okay, that is the division, that is the difference between studying a topic first time and a revision. Okay, so this is how, these are the tools that you can use for revision. And also for unit 1, there are a lot of numericals, so you cannot skip those numericals. So from where you can practice, definitely you can practice it from the previous year question papers. You can also take up our aim net series, which has all questions, previous year question papers, and it is divided in units. You can also try the questions given in the book of Biotecnika Stuffnet. Okay, all the toughest, it's a collection of very tough questions from all previous year CSIR paper only. Okay, so if you practice these questions, you will get enough exposure to how to handle and tackle this kind of numerical questions based on unit 1. Now let's move on to unit 2. And before that, there's an announcement for the Guru batch. So, on this month where we celebrate our Teachers' Day, we are launching our Guru batch for CSIR Net Life Science. And with the Guru batch, you are getting some freebies. Okay, you will be getting the online notes, classes, presentation, that is the regular stuff. Along with that, you will get the virtual internship on CRISPR. Plus, you will get our new collection of mind maps. Okay, during the session also we will show you some example of our mind maps, and this is also a very smart revision tool that Biotecnika is very soon going to launch, and whoever enrolls with the Guru batch will get this for absolutely free. For more information on it, please contact us in this toll free numbers. Batch starting from 5th of September. All right, so moving on to unit 2, which is about the cellular organization, or we often call it just like cell biology. So, what are the important topics here? Membrane structure and function, solute transport, FRAP, protein targeting, cytoskeletons, genome organization, unique and repetitive DNA, transposons, cell cycle and its regulation growth curve. Now, transposons is a topic which is also present in Unit 8. So, there's no need of studying it from both the units twice. Study it once and it is covered in the syllabus. Okay, the same transposon, whether you study it in cell biology or you study it in genetics, it's the same transposons. And as we have seen in the last paper, in the last paper that happened in February, a lot of questions came from transposons, so definitely you cannot leave that topic. Now, how to revise here? Okay, now unit 2 is kind of fact based, right? It's very theoretical. Okay, it is fact based. So you can highlight, you can use highlighting. Okay, but remember, because once we start highlighting, we often get carried away and we start over highlighting. Sometimes it happens that we end up highlighting 90% of the page. Have it happened with you? Like when you're reading, you feel like everything is important and keep on highlighting, and you end up highlighting 80 and 90% of the page. Have it happened ever? Okay, Harsha and Parumita, if the topics are not visible, you can just increase the you can change the picture definition. Okay. Yes, it happened. Yeah, tell you the truth, it happened with me also. Okay, so yeah, so how to avoid that? Okay, so when you are highlighting, you highlight with some annotation in it. Okay, annotation means where you add something extra. See, I wrote here because this thing is not written on the slide, so I annotated. Okay, so you write the extra point, something that you have learned in the class, something that you have learned in your college, something that you have read in some other book. Okay, so add that information also along with that. Okay, then highlighting helps because then you are recalling something, you are using your brain. Okay, you are using your brain to recall an information that you have attained some other place and then writing it down. Okay, so next time when you revise from those highlighted portion, it will be an added, okay, it will be an added information. Okay, so you use with annotation and avoid over highlighting. Next way how you can prepare unit 2 is by prepare to teach. Okay, see, you all have friends, you all do group study. Even if you are not doing group study, you just call 5 of your friends and say, okay, we all will prepare one topic and we will teach each other. Okay, so when you do that, when you explain a topic to a peer, so that times your brain is actively involved in recalling the information, also in conveying the information to others. Okay, now at that time your peers will have some questions, so you will be answering them. So that cross questioning will also help you in understanding the concept better. Trust me, it works, it helps. Okay, and in that time you will get to know, like if you are stuck somewhere, then your peers can help you, or you can look back in the book, you all can sit together with the book and refer it and try to find out the answer of the question that was bugging you in the session. Okay, so this is a very helpful strategy for all the topics which are kind of fact based. Okay, this is going to help you for sure. But definitely when we are doing group study, it is very easy to deviate from study and start talking about movies or weather or politics. Okay, so that also you have to stop. So no deviation, stick to study. All right. Okay. Now again, for the metabolic pathways, or for for the things where there is a sequence, okay, which comes one after another, definitely flow charts will help you. Like for protein targeting, like from where to where, from which organelle to which organelle the protein is going, how is it going, okay, which path will it take. All right, then for cell cycle and its regulation, definitely flowcharts are going to help you. What are the CDKs, what are the cyclines, how it's the phosphate kinase is working, how is it turning on or turning off a checkpoint. Okay, so they are going to help you in case of like this kind of topics where there's a sequence. All right, so flowcharts you can again make. Okay. Now, moving on to Unit 3, which is Fundamental Processes or Molecular Biology. So, this one is quite important from exam point of view. So, from DNA replication, important topics will be the Michelson style experiment, prokaryotic and eukaryotic replication, okay, telomere replication, etc. Then comparison of the different DNA polymerases. Then you can do from DNA repair. Definitely all kinds of repair pathways are important. From transcription, the transcription factors, RNA polymerase structure and types, post transcriptional processing, all the processing, okay, like splicing, capping, polyadenylation, editing. Then from protein synthesis, that is translation, okay, so there the steps of translation, the amino acid tRNA synthetases, codon, what is codon, anticodon, how do they pair, what are the post translational modifications, what is the function of each of the modifications. Then definitely one more important topic is this gene regulation in prokaryotes. So there the operon model, very, very important, and the various mutations, how it changes the operon structure, what will be the function at that if those mutations happen. And also in gene regulation in eukaryotes, where there is gene silencing, then the RNA interference. Okay, so these are the important topics from Unit 3. Now, how to prepare for Unit 3 or how to revise for Unit 3? Okay, so one trick that you can use is organizing information. Okay, so what you can do is whatever material you have, okay, or whatever text you have referred for studying Unit 3, arrange those notes and materials according to in the topic, okay, of the topics according to the difficulty level. Okay, whichever you find most difficult, okay, from there to the least difficult, okay, and accordingly you can color code, like most difficult you can code it red, little bit lesser difficult you can code it orange, like that you can, like whatever is the color code you want to prefer, you can choose that, color code the topics, okay, and then arrange them in that order of most difficult to least difficult, most to least, okay, like this. And then you start with the most difficult ones, okay, because the most difficult ones are supposed to take more time, least difficult ones are supposed to take the least time, right? So when you can afford some more time, start with the most difficult ones and slowly move towards the least difficult. Okay, next, in Unit 3, many graph based questions come, okay, especially when they are based on operon, when they are based on the DNA replication, okay, so this kind of graph based questions come. So definitely you need to practice those graph based questions. Again, for that you can refer to previous year question papers, or you can refer to our ToughNet book or any collection of previous year question papers that will help you in solving this question. So when you solve, then you will develop the practice of how to handle those questions. Okay, that is why solving this previous year questions are important. And also I told you important topic, one of the important topic is like comparative study of all the comparative study of all the polymerases. Okay, so how do you do that? In that case, make comparison tables. Okay, now here, and also for translation, transcription, replication, prokaryotic, eukaryotic, both are covered in your unit, so you can study both of them together if you make a comparisons table. Okay, so you will get to know what are common and what are the differences there. So in that case, again, Biotecnika has a very smart tool called as Conceptica wheel and Conceptica table. Okay, so this is the example of our Conceptica table. You can see here that in the same page we have got prokaryotic replication, eukaryotic replication. All important points have been noted down here. So you can easily have both, or you can cover both the replication processes at one page. Similarly, here we have the comparison between the different repair systems. Okay, so you are studying two repair systems at one time when you have this kind of comparison tables. Okay, so again, you can take help from Biotecnika or you can make your own table. Choice is absolutely yours. Okay, so before we move on to unit 4, yes, guys, how is it going on? Are you finding these tricks helpful? All right, Paromita, Mirinan, okay, Kushbu, good, 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 good. Good to hear that you people are here and you are finding it helpful. Okay. Then we have unit 4, which is about immunology and cell signaling. So, important topics, okay, which you cannot skip, you cannot afford to skip at the time of revision is signaling pathways, okay, then cell adhesion molecules, apoptosis, then cancer, everything related to cancer, then the drugs, anti-cancer drugs, then pathogen induced diseases. This is also a very commonly asked question in CSIR paper in part B as well as part C, okay, where they ask questions related to pathogens and what kind of diseases they induce. Then antibody structure and type, B cell, T cell activation, major histocompatibility complex, complement protein, cell mediated toxic cytotoxicity, as well as inflammation. Now, quick revision tips, tricks for Unit 4. Again, you can do it with flashcards. And again, there is one more tool that Biotecnika has, is called as Conceptica Clouds. Okay. So in Conceptica Clouds, basically, we have those important topics, okay, from a section or important words from a section in the same card. So you just look at the card, see the topics, and try to recall that in the brain, like what it was. So you can use these flashcards and concept clouds for genes involved in cancer, okay, apoptosis, MHC, cell mediated cytotoxicity. So this is how our Conceptica clouds look. Okay, so this is like a word cloud. All right, so as you can see, so this is apoptosis. So you see the word and try to recall immediately what apoptosis is. Okay, then hyperpolarization of rod cells. So you think, right? Then TGF, beta signaling pathway. So what is it? What it does? What happens in that pathway? Okay. Nuclear receptors. What are they? What do they use it for? Okay. So all these things, everything, you have this in the same card. Okay. Just have a look at the card and you have to revise the topics in your head. Okay. The next thing that you can use for your smart revision is the for the flow charts. Okay, you can make flow charts or you can make it in the form of a pathways, okay, where you have this again the events that happen in a sequence. Definitely you can note it down in a flow chart, like monoclonal antibodies or cell signaling. Okay, so this is a flow chart, as you can see from our Conceptica book, okay, Biotechnika's Conceptica flow chart book. And there you can see it is talking about monoclonal antibody preparation. Okay, so everything here is written. All right, so you have this pathway also is given here, and then with the help of image also it is shown what are the steps of monoclonal body antibody preparation, which again is an important topic. The another smart tool that you can use is foldable revision prompts. Okay, now what are these foldable revision prompts? It looks like this. Okay, just take a paper, cut it down in that way. Okay, so you can see these are the parts of circle it is mentioned. Okay, so what is circumference, what is diameter, like that. So definitely you don't have to do it for a circle or for any geometric figure, but you can use it for the topics like anti-cancer drugs and their action. So you just write the drug's name on the top and what they do behind it. Okay, so this will be very small also. So even before your exam, like if you have to travel to a different city, so it won't be possible for you to take all those heavy books or those large number of notes. So these tools, they will help. Okay, so you can take these small things with you, you can definitely carry it with you, and you can revise it right before the exam as well. Okay, so you can write the anti-cancer drug here and write the what it does behind it. Okay, so that will help you in revising. One more thing you can do in you can use this foldable revision prompts. They You can also use it for the pathogens and their diseases. Okay, so there are many pathogens, different kind of diseases they cause. So again, you can use this kind of revision prompts. They are very, very helpful. Thank you so much, Paramita. Thank you for your feedback. Okay, now moving on to unit 5, that is developmental biology. Okay, now developmental biology, the important topics are again listed down here. Okay, few important topics will be the different steps of gametogenesis. Then there will be a comparative study of different steps of gastrulation in chick, amphibian, C. elegans, and C. urchin. The different kind of genes that are activated in different pathways. Okay, in different kind of development. Okay, then slow block and fast block in polyspermy. Okay, and from the plant developmental biology, double fertilization, flower development, the genes involved in that. Okay, definitely important topics. So, how you can do a quick revision of Unit 5? So, you can make tables and flashcards for the basic terms and terminologies. Okay, so here you can see, right? So, this can be our this is an example, okay, of our cloud, okay, Conceptica cloud. So Conceptica cloud, you can use this like that. You can make flashcards. So you can write the basic terms, okay, and then you can revise it very easily. Next thing you can do is make comparison tables, okay, for the different gametogenesis, different stages of gametogenesis, or different comparison of gastrulation between in chick, amphibian, C. elegans, and C. urchin. Okay, how it happens. So along, if you make some comparison tables, so I'll give you some example that I have taken up from our Conceptica table. So this is Biotecnika's Conceptica table. So you can see here the fertilization in sea urchin and fertilization in mammal. So again, important things covered in the same page. Okay, so definitely the amount of book, the amount of text that you have to refer to is very less, and that will all help you in remembering the important facts, and they will filter out the not so important facts. Okay, now you can take, this is a Conceptica books content that I have taken for the different axis formation in frog, how it happens. So that is also given in the form of a flow chart. So flow charts will help you in revising the thing much faster, and because of the colorful images, you will have a better retention as well. So again, you can use foldable revision prompts just like I showed you in the previous one for the different genes and their function in the different stages of development. Okay, and flowcharts for all signaling pathways involved in the development. Next, moving on to unit 6, which is plant physiology. Okay, so the important topics: photosynthesis, plant respiration, hormones, and sensory photobiology. So, how to revise? So, definitely for this metabolic pathways like photosynthesis or respiration or nitrogen metabolism, you can use the flow charts. Okay, you can make it on your own. You can take the image of the flow chart from any standard book, or you can take it from our Conceptica book also. Okay, and see, this is the atmospheric nitrogen fixation steps that is given here. Okay, so just in a glimpse, just in a glimpse, the entire concept of nitrogen metabolism will, or nitrogen fixation, will be clear to you. Okay, now as I told you, with the guru batch you will get the freebies as our newly launched, yet to launch actually, the Biotecnika's mind maps. And this mind maps, I will show you some example of mind maps. So mind maps you can use for hormones. Okay, now what are these mind maps? So the mind maps will be all important things jotted down in a single uh, page. All right, so here you can see this is a summary of phytohormones. Okay, so there the list of phytohormones are given. What are the different signaling pathways that these phytohormones participate, that is given. What are the name of receptors that responds to these phytohormones, and what are the nature of the receptors that is given for these phytohormones. Everything is listed down on the same page. Okay, so just important things, nothing that you have to filter out here. Okay, so you can do it. You can create your own mind maps for the different biosynthesis processes of different hormones. And again, phytohormones are one important topic for CSIR exam. Then how they are stored, what is the mechanism of their action, everything you can have it at one place with the help of mind maps. Unit 7, again, animal physiology. So these two units, unit 6 as well as unit 7, they are very much very they are very fact based, right? You have to memorize a lot of things. Okay, so just blindly if you start memorizing, then it will become difficult for you to remember everything. So if you take help of the smart revision things, then only you will be able to memorize the things easier and faster. So again, important topics: blood clotting, ECG graph, oxygen saturation curve, action potential, the different sense organs, definitely. Structure of nephrons and its function, okay. Then endocrinology, reproduction, again, gametogenesis and ovulation. So revision tricks, you can make study starts. Now, what are the study starts? Okay, it's nothing very complicated, very simple, something like this. Okay, so you just take a paper and you can fold it up in different ways and write down the things on the same paper. Okay, just like I told you with the foldable revision prompts, this study starts are also it takes very little space. Okay, it takes a lot of small space, so you can carry it with you. Okay, definitely don't even try to carry it to the examination hall. Okay, that then you will be completely disqualified from the exam. I'm giving you this for your revision. Okay, so when you are doing your revision, you can use this kind of tools. Okay, so you can make study starts for blood clotting or the sense organs, their structure, their function, also action and function of nephrons. These are like these topics. These are the topics where there are many branches that you need to remember. So you can make a study start for that. Again, for mind maps, you can make mind maps and you can take help from Biotecnika's mind maps that will definitely help you in retention. So this is, you see, this is Biotecnika's mind map for female reproductive cycle. So can you see everything, every single thing about female reproductive cycle? Like when is the ovulation, when is the proliferation, what are the hormones involved in each of this step? Okay, everything is mentioned. All right, along with the image here. So you can refer to the image, you can refer to the text, and a big important topic is completely concluded in one page only. How is it, guys? How do you like the mind map? Do you think it will be useful for your revision? Yeah, we also think so, and that's why we are trying to help you, all the students, and making this mind maps. And it will be launched soon, and whoever enrolls with the Guru batch will get it absolutely free, the entire collection of mind maps of all 13 units. Okay, so again, you can make your own mind map or you can take it from the Biotecnika, completely your choice. Thank you, Pharma Medisol. I'm glad that you are liking the webinar. Okay, so before I sign off and pass it to Nidhi ma'am, just some general tips. Definitely, this is the time to buckle up and practice, 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 practice. Practice previous year question papers. You cannot leave that at all. Okay, so again, I will remind you of our ToughNet book, which is a collection of all difficult previous year question papers. You can take help from that, or you can take the AIMNet series, which will help you. Okay, and when you are trying on this AIMNet and all the questions, okay, when you are trying to solve the previous year question paper, make sure you sit with a clock. Okay, do not sit without a clock, because clock will help you in reminding how much time is left. You cannot spend 24 hours in solving 150 questions. Okay, you have to use your time. You have to be very, very economic with your time. Okay, so that you can solve all the required number of questions in the time given. Okay, all right, and yes, you keep on doing it. Okay, then your mind will be completely blocked. It will be like over information. So make sure you take regular breaks. But one thing that you have to remember, that you have to return to the revision. Okay, just break cannot last for half a day or whole day. Take break, take small but frequent breaks, but come back. Okay, when the break time is over, when 5 minutes, 10 minutes, whatever you are giving yourself after 1 hour or 90 minutes. Okay, 90 minutes, 10 minutes break, absolutely fine. Take the break, get refreshed, let your mind join the dots. Okay, let your mind have a thinking process which joins the topics. Okay, connects the topics, joins the dots. All right, and then you return to your revision. Okay, don't let time just go by that. Okay, you have to use your time very, very economically right now. All right. So thank you, everyone. Remember, every success story is a tale of constant adaptation, revision, and change. So today I tried to give you some important tips and tricks that will definitely help you in your revision. Thank you, everyone. And passing the control to our next speaker, Nidhi ma'am. Thank you once again, everybody. Nidhi ma'am, all off to you.
Thank you, sir. Thank you, Tithi ma'am. A very good evening to all of you. I'm Dr. Nidhi from Biotecnika. I would like to thank everyone there for joining in and being part of this webinar where we are discussing last minute advanced revision tricks for CSIR Net Life Science exam just before the exam happens. So yes, firstly, I would like to get the confirmation if I'm audible. Yes. Can you please confirm? Is my voice loud and clear for all of you? Good evening, Meera Rai. Good evening, Paromita. Okay, all good? Great. Great to see you all. Fine. So I'm sure by now you got an insight, right, that how we can prepare a smart revision strategy for units 1 to 7. I'll take it forward for units 8 to 13. But before that, I would like to ask one question here. Yes, what is the biggest difficulty we all face just before the exam? Think and let me know in the chat section. Yes, what is the biggest trouble that bothers us just before our exam? Thank you, Khushbu. Thank you, Fama, for confirming. Revise all the important topics in a day, Paromita. Okay. Stress. All right. Tension and stress. But why does this tension and stress come? What is the reason? Reason being memory erosion. Am I right? Memory fades away. And this happens to all of us. Let me tell you, this is natural, this is human thing. So firstly, stop cursing yourself, stop blaming yourself, right? Stop being guilty that why you are not only able to retain the things, or why you are not able to recall the things when you have learned all the concepts during your learning process, right? So we have scientific reasons for that, that memory erosion happens. Okay, but we can always think of some smart ways so that this process get reduced, right? The curve, the downward curve for the memory, right, that can be flattened, and that can be done with the repeated revisions, right? So spaced repetition, and the formula is LRR: learn, review, and re-review. I'm sure by now all of you might have gone through the first stage or maybe second stage as well, and now we have to do re-reviews. Let me tell you that when we actually learn anything, that goes, our brain stores it in the short memory, right? And if we don't retrieve that information very often, it will dump it. So keep revising, right? Try to retrieve the information after every regular intervals. Like now, when the days are less to our exam, so you can keep the time intervals like 2 days, right? You can revise after every 2 days, and then you can extend it to 3 or 4. So that will help you to have a better retention. Okay. Fine. So now let us move further to know that what are the quick revision tips for Unit 8. Right? How to do quick revision for Unit 8. Inheritance biology. This is one of the important units from CSIR syllabus. As we all know that we cannot imagine any CSIR exam paper where you won't find any question from Unit 8. Am I right? Yes? Okay, so let us see how we can quickly revise the important things from Unit 8, right? So foremost, all the genetic terminologies, they should be at your fingertips. So revise all the important terms from genetics, right? So how we can do it in an easy way? Refer glossary, right? Refer glossary from the books. So there you will find all the terms with the simplest descriptions. Okay, no matter, let it be a very simple term. It's a chromosome, whether it's homologous chromosome, it is gene, right? Difference between gene, allele, gene locus, gene loci, homozygous, heterozygous, dominant, recessive. What is the difference between character and trait? Or the other terms like codominance, incomplete dominance, pleiotropy, genomic imprinting, then penetrance, expressivity, phenocopy, linkage, epistasis. All these terms are important. Okay, now different books will do it in different ways, but this is the simplest way to revise all the terms. Am I right? You will find all these terms at one place, and that too with simple descriptions. So definitely this will help you to revise all the important terms from genetics. Okay, Paramita, great to see that how you are having a strategy to revise the things just before the exam. Good to see that you are working on it actually. Now, let us move to our next slide and let us have a look at what could be the other approaches to revise the other important concepts from Unit 8. Definitely from Unit 8, you can expect numerical based question. Am I right? Now let them be the probability sums based on Mendelian ratios, or it will be from gene mapping, or that is numerical type questions from pedigree analysis, or whether that is chi-square test. So yes, you have to practice on questions and problems, right? Where you're going to find these questions and problems at the end of the chapters, right? In these standard books, you can refer these standard books, or if you are having Biotecnika material, so that will definitely be helpful. Okay, so if I just give you some reference books for genetics here where you can find these questions and problems. So here we are, Russell, Pierce, or Sinested. Okay, now you have to solve the problem on your own, right? And then match your solution to those given in the books. This is the best way to revise the formulas. Trust me. Okay, when you will match your solution to that which has been given in the book, you will be able to find out that where you are going wrong, and then it will be the best time that you would be able to revise the formula related to it, and you are going to retain it for long, right? And then don't forget to refer the concepts, keynotes, key points, basic exercises, testing your knowledge, summary points, connecting concepts, analytical approaches which are given in the books, right? All in all, the summary points, the highlights which are given at the end of the chapters, or maybe somewhere in the middle of the chapters, they are really important, and this is the best way to revise all the important concepts from a particular topic. Yes. So I hope you got an idea that what I want to convey from this slide. All right, now let us move to our next slide. Yes, we can always use smart tools, right? So Titi ma'am has already talked about that how you can use the smart tools to make your revision process easier, right? So like if I talk about comparison tables again, comparison is something when you're going to compare one thing with another, so it will help you to memorize and to retain that thing for long. Okay, like again, to give you an example here, addition rule and multiplication rule, right? We cannot ignore these two rules because the questions they are asked based on them in the exam, right? So why not to use in comparison tables here? All the things they have been summarized, and that too in a comparison form, so it will be a quick revision for us, for revising this topic or any other topic in this form. All right. Now, another example, like here we can see the comparison between genetic mapping and physical mapping that has been summarized. So these are the screenshots, right? These are the example from Biotecnika concept tables, right? You can create your own comparison tables, definitely, right? But that would be time consuming. So you can always reach Biotecnika if you are interested to have these concept tables with you. Yes, so let us talk about the topics which you shouldn't skip when you are revising Unit 8. That means the important topics. So firstly, we'll talk about Mendelian ratios. Yes, Mendelian ratios, you need to revise all the ratios. They should be on your tips. Okay, and if they are related to the extension of the Mendelian principles, that is also important. So the best way to revise these ratios is again to do it in a tabular form. Okay, so if you already have some tables with you for that quick revision, that's well and good. Or if you can create it quickly, or again you can reach Biotecnika if you want us to help you there. Okay, so this kind of table that is going to help you to have a quick revision over these ratios, because again you can expect part B and part C questions to be asked based on probability sums, right, related to Mendelian ratios. Next, gene mapping, an unskippable topic. Definitely one question would be there in part C. Okay, so gene mapping, how you can revise it. Take one example, write down the entire method, right, in step to step, and then you can solve the other problems by taking this example as a reference. All right. Then interrupted mating experiments, microbial genetics, right? HFR mapping, different ways for gene transfer, transduction, conjugation, transformation, right? Now, how we can have a quick revision over these? Again, you can use these tables, right? So this is one example I have taken from one of the standard books I have shown to you, right? So here you can see the expected results from the mating experiments, right, for conjugation that has been summarized. Pedigree analysis, as all of you might be knowing, either part B or part C question you will definitely be getting in the CSIR exam. Now, how you can revise pedigree analysis? So there again, take help from the diagrams or from the flow charts. Okay, you need you need to ensure that you are well familiar with all the symbols, because if you do any mistake on that, so definitely you'll not be able to get the right solution for pedigree analysis. Mutations, another important topic, students, right? Let it be spontaneous mutation, induced mutation, suppressor mutations. So you can revise mutations in the tabular form, comparison tables, or Biotecnika's concept wheels, right? Chromosomal alterations, so they are deletions, duplications, translocations, inversions, or ploidy, right? So all that you need to revise. Tetrad analysis, complementation test. So all these topics, they are very often asked in the exam, so please don't forget to include them when you are revising with unit 8. Thank you so much for your feedback, Sharon, Mirinon, Paromita. Thank you so much for your feedback. We really appreciate your comments here. All right, now let us move to our next unit, Unit 9, Diversity of Life Forms. Right, if you have a good idea over this unit, right, and if you are good in memorizing the thing, so you shouldn't skip some important topics from Unit 9. So generally part B questions, they are asked based on terminologies. Now, if you'll ask that what kind of terminologies they are asked, okay, like cladogram or phylogram, or let it be monophyletic or paraphyletic, or it is ureotelic, uricotelic. So questions related to them, they could be asked in part B. So again, you can revise these terminologies from glossary. Okay, so that would be the easiest and a quick way to revise all these terms. Then, if we talk about part C questions, so usually match the following type questions they are asked, and that too based on common parasite and pathogens for different hosts, different protozoans, and bacterial diseases. Now what is the best part about Unit 9 is that you can club the topics to other units for revision. Yes, see, the topics like rare and endangered species, conservation strategies, major habitat types, they can be clubbed with Unit 10 for a quick revision. Right? Adaptive radiation, adaptive modifications, evolutionary relationship among taxa, migration of species, club them with Unit 11. Parasite and pathogens of humans, club them with Unit 4 revision. So this is how you can make the revision for Unit 9 topics quick and easier. Okay, so I hope that will help. Now, Unit 9 is mostly theoretical, right? Here, what do we have? We have hierarchical taxa, we have domains and kingdoms, we have comparative anatomy, and then we have the criteria for the classification, 5 kingdom classification, classification for plants, animals, bacteria, fungi, right? So, whether we will be going back to the books to revise all the things again and again, do we have that much time? A big no. Okay, so the best way to revise is, again, use concept wheels or whatever diagrams you have for now, okay, or the comparison notes to revise basic classification of animals, plants, bacteria, or even fungi. Are you getting on this? Right? And when you revise on this concept, focus on the characteristics because those are asked mostly in the exam. Cladistics and phonetics, you shouldn't be skipping on this. Different types of classification system, diagrammatic notes for correlating plants and animal groups, know the phylogenetic tree and should practice questions based on it. Now, see, I'm sharing one more example here. Now, let us suppose you have to revise characteristics of gymnosperms. Like I said, right, we don't have time to go back to the book and find out all the characteristics from point to point. Okay, so the best way is revise all the characteristics from a diagram. Like here, I'm sharing one example, which is Biotecnika's concept wheel. So as you can see, all the characters, they have been summarized and that too in a very colorful way. Okay, so definitely this is going to have a long-term impact on our memory. Or you can use comparison table. So that is always an easy, a simple, and a quick way to revise over the things. Similarly, you can have concept wheels for the characteristics of pteridophytes, bryophytes. So that is another way to quickly revise over these otherwise boring topics. Am I right? Okay. Now let us move to our next slide, and here I have another very interesting approach, and that is using mnemonics. Right? What could be better than using mnemonics to remember, like for unit 9, if I talk about this? So we can use mnemonics to remember the taxonomic ranks. Let me share one example if you know about this. Dear King Philip came over for great spaghetti, soup, or sweets. Whatever you like eating, you can mention it here. Okay, so the basic idea is that you can use a phrase which is very vivid, which is very interesting. Okay, and as you can see, the first letter that is referring to the taxonomic ranks here in our example. Okay, so in this way you can create mnemonics on your own, of your own, right? Whatever you like, you can use that to create some interesting mnemonics, or you can even use short rhymes or stories. So isn't it, it's a creative idea to revise the things? Yes, can I get some feedback? All right. So yes, meanwhile, let me move to our next slide. And if you have missed on this, so let me share it once again. We are coming up with the Guru batch starting from 5th September. Okay. So Guru is the person who enlightens our life in multiple ways. Like, so this is where we are starting with this Guru batch. And apart from the regular things Biotecnika always offers, you are getting complete mind maps of the entire CSIR net syllabus, as Tithi ma'am has already shown you some glimpse of the these smart tools. Yes, mind maps in her presentation. Then smart sync timetable, Guru assignment per student, presentation, online notes, and daily practice sheet. And yes, you are getting CRISPR virtual internship as a freebie with the enrollment for Guru batch. So what are you waiting for? Call us at our toll-free number, or you can connect to Biotecnika. So there are different ways to do that, as we have shared the information on that, right? So now let us move to our next slide, and here we have unit 10. Yes, ecological principles. Okay, Mirinon, good to see that you know about this mnemonic. Okay, you saw it in a TV show. All right, great. Okay, so now let's talk about unit 10. This is again an important unit, and what are the topics which should not be missed during your revision? Habitat and niche, and all the terms related to niche. Again, you can refer glossary, right? Always remember that is a very that's a very simple way to have a quick revision. Refer the glossaries, but yes, you must refer some standard books or standard source material when you are doing that. All right. So terms related to niche, let it be fundamental niche, realized niche, niche breadth, or whether that's competitive exclusion, whether that's resource partitioning. So all the terms should be clear with you all. Next, R and case selection, always asked in the exam, right? Whether in part B or part C, you'll always expect a question based on this concept. Then we have survivorship curves, population growth curves, right? Again, you can expect numerical based question from this topic. Meta population concept, and a very important concept, right? Species interactions, practicing numerical on population growth models. Yes, we cannot skip on it. Okay, so you have to cover both the population growth models, exponential and logistic. Then from ecosystem ecology, net and gross primary productivity, secondary productivity, this all you need to cover, right? Now, mostly from unit 10, diagram and graph based questions they are asked. So you have to practice graph based question, right? You need to learn that how to analyze and graph, how to reach the right answer, right option in reference to the information given for the graph. Okay, now what are the topics from where you expect the graph based questions to be asked? Lotka-Volta model, predator-prey relationships, theory of island biogeography, species diversity, top-down control, bottom control. And trust me, all these topics are very simple. If you have already learned them, I'm sure you might be knowing on this. Okay. So here I'm sharing one sample question, right? This is a CSIR question based on Lotka-Volta model. So like you can see here in the graph, a graph has been given and it has been asked that based on the information given in the graph, right, the carrying capacity has have been given, and looking at the pattern, you have to tell that what will be the predicted outcome of the competition between two species, right, that are having carrying capacity K1 and K2. So you need to learn, okay, and if you have already done it, you need to revise. So for that, you need to practice on these types of graphs. So revise from diagrams, graphs, flowcharts. Okay, so again, if I take example for Lotka-Volta model, if you'll decide to go, back to a book and find out or to revise all the information related to Lotka-Volta model, it will eat away your entire day, or maybe at least half of it. Okay, so the best way to revise this concept is again from a colorful diagram. Okay, like this is one example from Biotecnika concept wheel. As you can see, all the information along with the formulas that has been provided in the concept wheel only. Okay, or you can revise from the graphs. Now this is a screenshot from Biotecnika Conceptica book. Okay, so here as you can see, the graph has been provided. Both the models, exponential growth, logistic growth, the formulas, the curves, and then how they are related with the R and K strategies. Like I've already mentioned, R and K strategy, that is very important topic which is very often asked in the exam. So this will make your revision easier, right? And let me share you another example for mind map. Okay, so if you're gonna enroll for Guru batch, so you'll get these mind maps as freebie, right, with your enrollment. So just have a look at this example. This is the mind map for fundamental and realized niche. Okay, so you can see here the entire information, right, including the hierarchy of the terms. Like we'll start with the ecological niche, then we need to know that what is fundamental niche, what is realized niche, example, what are the important facts related to these terms, as well as diagram. Everything has been included in the mind map only. And it's not like that mind maps, they need to be boring, right? They can be colorful and you can add images to them, right? So let me tell you that there are free apps available, so you can use them to create your mind maps. Okay, but since now we are left with very short time before our exam happens, so if you can get these mind maps already prepared, and that too to the expertise, so I'm sure that will be really helpful to you all. Yes, so I hope everything is clear so far. All right, so let us move to our next slide and let us talk about Unit 11 now, evolution and behavior. So some topics from Unit 11, they are conceptual, okay, and some need to be understood, some need to be memorized, and from some topics you can expect the numerical type questions to be asked, right? Like you will be starting from the terms again, so you can refer the glossary, and then moving further to phylogenetic tree, then geological time eras, then Hardy-Weinberg principle or law, so numerical based questions related to that. Okay, and then evolution of behavior. So these are some of the important topics. Now how we can use the quick revision tips to revise these topics. Okay, so firstly, using mnemonics to memorize the eras, to memorize the periods, to memorize the epochs and the events which have happened during these eras, periods, or epochs. You can again create some interesting mnemonics on your own. Okay, or use flashcards. So Titi ma'am has also talked about flashcards. Okay, so trust us, flashcards, they work as a flash of light. They ignite our memory to respond quickly. Okay, so you can always use some colorful flashcards to revise the terms and the concepts. topics like lemma concept, Darwinism, adaptation, concept of operant Holden, to have a concept clarity between founder effect, bottleneck effect, Hamilton's rule, speciation. Right now, these are again some of the very important topics which are very often asked. Right, speciation. So here you have allopatric, sympatric, then evolution of behavior. So from there, kin selection, altruism. So these are again very important topics. Right, terms like adaptive radiation. So this is also related to Unit 10, endosymbiotic theory or types of evolution. Let it be convergent, divergent, or it is stabilizing, or it is directional, or it is disruptive. So all these terms you need to revise. Okay, so how you are going to do it quickly? So use flashcards, right? Or you can use Biotecnika's concept cloud. So here again you will be able to find out, all these terms at one place, and you can look at any of the term randomly, and whatever is coming in your mind, so that will help you to revise. If somewhere you will feel that, okay, I'm not able to recall this term properly, so you can go back to the glossary or to your standard reference material and you can re-review it, right? Use flowcharts to revise. Phylogenetic tree, cladogram, molecular evolution, molecular divergence, and biological clock. All right. Thank you, Paromita. Thank you, everyone, for confirming. All right, so let us move to our next slide. And yes, here again, Hardy-Weinberg law. How we can ignore it? Never, right? So you definitely will get some question based on it, right? So how to revise this law? Again, you can take help from a colorful diagram or a concept wheel, right, where you have all the important facts related to this law along with the formula. Okay, so and apart from that, definitely you need to practice on the questions. Okay, so keep practicing on the question whenever you practice on a question. And here also you will find some questions and problems in the standard books. So solve those questions, match your solution with that given in the book. Okay, you can also reach Biotecnika if you are interested in trying MNet series. Okay, or the ToughNet. So there you will get the toughest questions to practice on, right? So that is again a way to revise the things efficiently. Right, now let us talk about Unit 12. Yes, Unit 12, Applied Biology. So here you will find, mixed topics, right? And the questions which are asked in the exam, they would be statement based, or match the following type, or graph based. Okay, and direct questions, they are asked from agrobacterium mediated gene transfer, right? So this will be related to some transgene experiment, to the gene expression. So that is how direct questions are asked from this part. Now apart from that, other topics like molecular markers, biodiversity, from here you can expect match the following type question, or even statement based, right? And a good thing is that this can be merged with Unit 10 as well. Somatic hybridization, gene therapy, transgenic plants and animals. So in the recent exam, you had a part C question based on transgenic animals. That was a 4-mark question. Right, now what are the helpful tips which will help you to revise the things easily and quickly? So you can use comparison tables for gene therapy. Okay, like whether gene therapy that is somatic, that's germline, that is in vivo, or whether that is ex vivo. So use comparison tables. Molecular markers, RFLP, RAPT. So there again you can revise the things using molecular these comparison tables. Okay, flow chart for agrobacterium gene transfer. So that is the best way to revise that how agrobacterium mediated gene transfer technique that is used. Okay, mnemonic for remembering VIR genes of TI plasmid. Okay, and then we can always club these topics with other units, like club with Unit 13 topics like molecular markers, or somewhere you will find the techniques from Unit 13, they will be merged with the transgene experiment. Okay, like Southern blot or ELISA or RIA. So these techniques, they'll be merged with the transgene experiment. Club with Unit 4, vaccines. So you can always club this topic with unit 4 to have a quick revision. Then some topics they can be clubbed with unit 10 as well, like I have already mentioned, biodiversity. Okay, so the good thing about unit 12 is that you need to cover only some topics from unit 12. Okay, CSIR aspirant should be covering some topics from unit 12, and then you can do it in parallel with the revision for other units. All right, so coming to our last but not the least, unit 13, methods in biology. Very important unit. Okay, 12 to 14 questions you can expect to be asked in the CSIR exam. Okay, so yes, what is the first tip I would be giving you? You need to revise with all the formulas. Right now, you can do it by using the conventional technique where you can list down all the formulas on the paper, right, with proper labeling that what all these variables would be representing. If you already have that list prepared with you, so that would be really helpful. Okay, and the other advanced way to do that is record all the formulas. Okay, record all the formulas in your own voice on a phone, and you can listen to this recording later as many times as you want. So that will, serve the purpose for re-reviewing. Okay, so we need to re-review the things. Remember LRR formula. Okay, so this is how you can listen to your recording again and again, right? So this will be really helpful. And whenever you will be solving questions based on these formulas, give yourself a target, right? Like 3 minutes, 2 minutes, 4 minutes, and try to solve the question within that target time. Okay, so if you find yourself unable to do that, again, you can go back to the formula, right? So, revise it properly, find out where you are going wrong, and then you can retry. All right, so this will be helpful. Then while learning the technique, focus on principle and applications. Don't miss on recombinant DNA technology. And there you need to focus on manipulating and modifying enzymes, restriction digestion vectors, very important topics. I'm sure you might be having an idea on this. Okay, so you need to revise these topics for sure. Then flow cytometry, UV visible spectroscopy, radioisotopes, and half-life or microscopy. So from here, you will definitely get part C questions, and that too mostly graph-based questions they are asked. And yes, use smart study tools. They are always helpful, right? We need to revise smartly, right? And therein, these smart study tools will help us. Okay, so let me give you a gist that what are the important formulas from unit 13 which should not be missed while you will be revising. Okay, so here you can see probability of restriction site with non-ambiguous sequence or with ambiguous sequence, Beer-Lambert's law, protein molecular weight determination by mass spectrometry, chi-square test, degree of freedom, Poisson distribution formula, confidence interval, radioactive decay, half-life, microscopy, that is Ernest Abbe equation, it's important, right? Polymerase chain reaction. So like this, you can create your own list and re-review that list as many times as you can, okay, to remember and to retain these formulas for long. Yes, revise from comparison table, flowcharts, wheels, diagrams, mnemonics. Again, to revise the techniques, you can take help from some creative mnemonics. Okay, and here for revising unit 13, animations will be a great help. Okay, if possible, watch animations for the techniques, so that will help you to revise a technique quickly. Okay, and bring variation to the source material. Unit 13 is huge, right? You need to revise a lot of things from unit 13. So there, bring variation to your revision material, right? You can always switch from comparison table to flowchart, or from flowchart to wheels or diagrams, mnemonics, like that. Divide the techniques into small chunks to remember. Okay, like if you are learning a technique, divide first three steps or keep those three steps as one chunk. Okay, and you can again add some kind of mnemonic to it to make it interesting, and then the next three steps you can revise it by using another mnemonic or some other way. Okay, so in this way, just try to break up into small bits, and that will help you to have a efficient revision. Okay, and yes, comparison table, they are always helpful. Like if I tell you about this, one of the important topics, restriction endonucleases, so you can find all the details for all the three types of restriction endonucleases at one place. This is again Biotecnika's comparison table, which I'm showing you here, concept table. Okay, so you can revise the things in this way. Then flow chart, like this is an example, okay, for RIA technique. So this can again be helpful to revise a technique quickly. All right, so this is another example, which is a mind map on restriction enzymes. Okay, this is just a screenshot of it. So here, as you can see, restriction enzyme, three types. Okay, application, classification, and yes, before that, introduction. Okay, so this is just a part of it, right? And the other things, like you need to know about the blunt ends, sticky ends. So the entire information will be available in one mind map. This is one of the Biotecnika's mind map I'm showing you here. Okay, so this is how, mind maps, they are very, this is one of the interesting way where we can link the ideas with one another. Okay, and here in our case, we can connect the terms and processes with each other to have an efficient revision. And you can always make them colorful. You can add images and the other things to make them look appealing. Okay, now while revising unit 13, try to relate while you revise. Okay, how you can do that? Club it with other units, like unit 13 and other units like units 1, 4, 8, and 12. Unit 13 can be clubbed with unit 8 and 12, like we had seen the topic molecular markers, it is common. Then unit 13 and unit 1, radioactivity, this is something common, right? Unit 13 and unit 8, statistical methods, binomial distribution, chi-square test. So these are the topics which you can revise quickly by clubbing unit 13 with other units. Now, I have another technique for you, and this is where we can read, cover, remember, and retell. If you are able to recall, this technique we used in our childhood. Remember? So, that part of your book which you can cover with your hand, right? So, read that much part and then cover with your hand. Okay? And then remove your hand and try to remember. Okay, or means if just read one paragraph, keep your hand there. Okay, and then try to remember what you have read in the previous step. All right, and then retell it to your study partner or maybe to yourself. So in this way, this will help you to revise the concept. Okay, you can repeat this exercise, just read one paragraph, again cover it with your hand, try to remember whatever is coming in your mind, right? And then you can retell it yourself or to your friend. Okay, now the next technique which I'm going to share with you is reverse revision technique. Right, now what do we have here? This is a very fun way to do it. Think like an examiner, right? Think of any term or its definition, and then just imagine what are the possible ways to create a question to which this term would be the answer. Okay, so here you have to think like an examiner. What is the question to this answer? So if we'll be using this reverse revision technique, so this will help you to revise all the facts, right? All the important things related to that term or its definition, right? And don't forget to reward yourself for every accomplishment. Yes, you have earned it, so why not to celebrate it? Let me tell you that your brain, it responds positively, it generates positive emotions, right, when you reward yourself for every accomplishment, because it comes to know that yes, every accomplishment that is being rewarded. So like you can eat something which you like the most, you can just go and hang with your best friend, or you can just watch your favorite TV program. Okay, but yes, definitely you should try doing this. This will definitely bring an internal motivation to you. So with this, I would like to conclude. I would say that don't make studying harder than it should be. We are living in an era where we have all the advanced technology tools with us. So try using these advanced tools to make the revision process simpler. Okay. Revise the things from the flowcharts, diagrams, concept tables, or wheels, like we have given, we have shown the examples, or the mind maps. Okay, and flatten uh, your memory curve, which usually uh, you know uh, where our memory fades away, so we can flatten that curve using these smart tools. So all the best to all of you for your exam, for your preparations, for everything. Thank you for listening to me.
All right, looks like we have just finished our session. How was it, guys? Please tell me in the comment section, in the chat, live chat. How was it? Did you like it? Yes? Okay, wonderful. Great. So now that we have come to an end of the session, I have a small announcement to make. On 5th of September, we are celebrating Teachers' Day right here on YouTube with all of you. And we have lined up some amazing surprises for all of you. So don't forget to attend the 5th September session. Okay, it will be 1 hour session in between 7 to 8. Please spare out 1 hour session, 1 hour time to watch this session, and you will get some amazing gifts from our teachers. So are you ready? Please don't forget, 5th September evening, 7 o'clock. It's a Monday. Thank you so much for watching this webinar. I'll see you in the next video. Till then, please take care. It was wonderful interacting with you. See you there. Bye-bye.